hydroxyl group, NH2, or a is it a
Are you talking about the amino
oxygen?
acids or the substrate?
Substrate.
So on the substrate, you have, I see an oxygen, I see an OH group, I see an NH3 plus group, and I see another oxygen double bonded, an oxygen with a minus charge. So you got several things here. I see a nitrogen. You got several different groups here. And basically, any group that's got an electronegative element is probably going to be polar to some degree, but the difference is when you have an electronegative element with a charge on it. So if something is a minus charge and a positive charge, that's not just polar, that's now ionic. That's so polar that the charge has fully separated. So this goes back to really fundamental content from chapter 4 of our textbook. But you have to make a distinction between groups that are just polar and would have, I'm going to share my screen. Need to plug up. Okay, you can see my screen now. Okay, that's stuff I wrote earlier today. All right, you got to make a distinction between, okay, so you're looking for a few different kinds of groups. Polar groups, well, first we'll just start with nonpolar. Nonpolar groups, and I'm talking about your substrate right now. You're looking for nonpolar groups because you know that what kind of amino acids, we've got different kinds of amino acids too. We've got specifically, if you're looking, let me just pull that, where is it? Right here. Nope, I wanted to do this, split screen. Okay. We are talking about proteins. If we're looking at amino acids, everything I've circled, we've got nonpolar amino acids with neutral side chains. We've got polar amino acids with neutral side chains. Then we have ions that are so polar they're now ionic. So I would consider these, like, if I could write on this, ionic. Okay, we've got some with negative charges, some with positive charges. Now technically we call these acidic or basic because the charge is there based on what has been given away or received. Things that carry a negative charge, like this right here, it's negative because it gave away a hydrogen. What it used to look like is this, okay? We call that aspartic acid. But when it gives the hydrogen away, now we get aspartate and it has a negative charge. That's acidic. Things that give hydrogens away are acidic, okay? And same idea with glutamate. Over here, arginine, lysine, histidine, these all can receive another hydrogen onto their nitrogen and they have a plus charge on this nitrogen, okay? If you draw that out, that nitrogen has 4 bonds, 2 to hydrogen, and then 2 to the same carbon, okay? These carbons are the same carbon, but it's just that I'm just drawing this out. So you see 4 bonds, there's gotta be a plus charge on this nitrogen, okay?
Right.
So it'd probably help you out if I just drew this as a double bond, okay? But it's received a hydrogen, so it's basic in character. That's why we say basic right here, and that's why we say acidic right here. So you are looking for things that can interact with these. So what kind of groups can interact with nonpolar side chains? Nonpolar groups, okay?
Right.
Nonpolar like dissolves like, nonpolar mixes with nonpolar, nonpolar is attracted to nonpolar. What kind of groups can mix with things that are polar? Polar groups. Why is that? Let's consider arginine for an example. This carbon right here, the carbonyl carbon, is bonded to 2 electronegative atoms, nitrogen and oxygen. These, whoops, I thought I was on. This oxygen and nitrogen, they are electron withdrawing. They are going to attract all the electrons in these bonds are going to be attracted towards the nitrogen, attracted towards the oxygen here, because these are electronegative elements. They like electrons. They're attractive to electrons. So that makes, it doesn't go all the way, but they're not shared evenly. These are covalent bonds in this organic molecule. The electrons between the carbon and the oxygen, between the carbon and the nitrogen, they're just not shared evenly. So that leaves this oxygen a little bit electron deficient. So it would have a partial positive. Partial because it's not a full ionic charge like down here, okay, like this or like that. These are full ionic charges. They're there to stay. unless pH environment changes or something crazy happens. But these are only partial positives, and then this oxygen would be partial negative, the nitrogen would be partial negative, okay? So again, you see a separation of charge. It's the same idea, it's just not a permanent full ionic charge. It's just a partial separation. So why can this interact with another polar group? Well, if we had cysteine here, well, cysteine's not a great example. Let's say we have tyrosine. Tyrosine has a side chain that looks like this. It's got an aromatic ring. Okay, that would be to the backbone. And then it's got this OH group right there on the end. This was tyrosine. Okay, well, this oxygen here, I'm going to go to a different color, is also partial negative, and the hydrogen is partial positive. Okay, so when you have other polar groups, two polar groups near each other, what happens is this partial positive sees the partial negative on the neighboring oxygen, and you get an attraction there. The same way that you get with ionic things down below. It's just not as strong. Ionic charges and ionic attractions are the strongest because you get a full positive and a full negative charge. When they see each other, they're highly attracted. Here, it's the same basis. Separation, you got a positive and a negative, and they're attracted to each other. It's just not as strong, but it's the same fundamental thing happening, okay? So that is why polar groups on substrates are really attracted to polar groups, side chains on amino acids. So if you're trying to analyze, okay, what kind of amino acids are going to interact with my substrate? Do I have any polar groups on my substrate? If I do, man, polar amino acids are going to be highly attracted to those polar groups. Okay, so that's just an example with polarity.
So
Keep
the
going.
carboxylate group and the NH3 positive, those are both ionic.
Yeah.
So I don't have to write polar or non-polar for those. I put ionic.
Well, I just use this as an example. I want you to use acidic or basic. The way we refer to these amino acids is we say, okay, aspartate, glutamate, these are acidic side chains. After they give their hydrogens away and act like an acid, they're left with a negative charge. Okay, but there's reasoning for why this course is not really all focused on acids and bases. That whole chapter is a preceding chemistry course. But with that understanding, that's why these amino acids are behaving the way they are. So we call them acidic amino acids or basic amino acids, but they have ionic character to them because of the charges that are present. So I would want you to say acidic or basic.
Okay.
And think about that. What specifically is going to be bonded or attracted to the negative charge? What specifically will be attracted to the positive charge? Don't say acidic and basic for both, because that's not the case. Remember, opposites attract. So if I had to pick, we've got 5 amino acids at the bottom of this table. If I had to pick one to interact with, this negatively charged oxygen right here, which one would you pick? There's more than one acceptable answer.
I can't see which one are you talking about the one on the left? Oh.
If I had to pick something to interact with this negatively charged oxygen right here, what amino acid would you pick from this bottom table?
What amino acid?
Yeah, so your options are aspartic acid, glutamic acid, arginine, lysine, or histidine.
Arginine.
Okay, why did you pick that?
Because I saw 2 positives on nitrogen.
Right, and remember, I told you guys, don't pay attention to the backbone. I just want you to look at the side chains. But you just told me, for what you said, you see a positive charge on the side chain.
Right.
And you think, all right, that positive charge is probably going to be attracted to the negative charge. And you're right. So arginine's not the only answer. Lysine could also. Okay, so the type of amino acid that's going to interact with this particular group right here, I'm on freaking eraser, would be a basic amino acid. See, we call these basic amino acids.
Right.
Does that click at all, Cameron?
A little bit better, yes.
Abby?
Yeah, would you want us to say that they react with basics, or would that be salt bridges? Or when the
So
salt-
when it's asking you, the question is what type, if I could ever get this thing, okay, what type of amino acid? So your types of amino acids, your options are acidic side chain amino acids, basic side chain amino acids, polar amino acids, or non-polar amino acids. Those are your 4 options. I also told you guys, if you wanted to go ahead and be even more descriptive, that's fine. You can tell me, you know what? I think a basic amino acid would react with this group probably through a salt bridge. Okay, the salt bridge part isn't actually answering the question, it's additional information, but it's really good to think through that.
Okay, thank you, gotcha.
Yeah.
Okay, question.
Yeah.
Okay, so now that I know what's basic and acidic, how do I figure out when it's polar or non-polar?
What are you talking about? The substrate, like groups on the substrate?
Yes, because I thought what the question was asking, we'd tell its charge, the polar groups, and what kind of side chain, right?
So, sorry, hold on, it locked me out. Okay. So yes, Cameron, you need to be able to look at a molecule, a substrate in this case, and identify, y'all can't see my screen, can you?
No, ma'am.
It's like it's stuck. Why is it stuck? I can see it on my iPad, it's not. Hold on, let me disconnect. Let me stop share and reshare. Come on. I mean, if I share to my desktop, y'all can see that. Yep. If I share to my iPad, y'all should be able to see that.
Would this be like most of the exam or no?
It won't be most of the exam. Why? Can I unplug? I'm gonna replug it. No, still nothing.
Nothing.
What the heck? All right, let me see if I can answer your question without. Let me just stop share. So, you need to be able to identify any sort of functional group that is polar, any sort of thing that is non-polar, anything that is ionic in character, meaning it carries a positive and negative charge. So,
Okay.
so how do we tell when something's polar or non-polar?
That was my question.
Okay. So, there's gonna have to be some backtrack because that is something straight out of chapter 4. So let me find the pages. Actually, I could just share. Well, if I can't share my screen, then I can't. I'm trying to find a. Let's see right here. Okay, here it is. Covalent compounds, right? No, molecular compounds. Here it is. Like, yep. Share. Now, hopefully I can screen share. Come on, boom, there it is. Okay. Cameron, I gotta scroll down. So this is all stuff that I go through in my previous chemistry course, the prerequisite to this one.
Mhm.
So that's why you haven't seen these slides. All right, right here. So I discussed in my previous course the idea of polar bonds and then the idea of polar electronegativity. Have you ever heard of like the electronegativity chart? Okay.
Yes.
Electronegativity difference. This is the key. So this is how we measure how polar a bond is, all right? So then I'll walk you through how to calculate electronegativity difference. And it all just goes back to the numbers on this chart. You can see that the closer you get to fluorine, the numbers get bigger and bigger and bigger. So everything over here, metals, okay, hydrogen's an exception. Hydrogen's not a metal, but your metals have very low electronegativity. They do not desire electrons. Electronegativity is a characteristic that it alludes to in a bond, how much does an atom attract electrons and pull electrons towards itself, almost like tug of war. Okay, so the difference is this: if you have two identical atoms, let's say we have 2 molecules of oxygen in a bond. We know that every bond is 2 electrons sitting in that bond. Now they're moving, but we draw them sitting in the bond. Both oxygens have the exact same nucleus. And what kind of subatomic particle is in a nucleus? Protons, neutrons, or electrons?
Sorry, Robin said, can y'all hear anything? And I didn't hear you.
Oh, okay. What, can you hear me, Robin?
Yeah, I can hear you now. When did y'all start?
Oh, Cameron asked to meet a little bit early, so we were meeting early for that reason. I'm not officially starting until 2:30.
Okay.
But we're going over bond polarity right now. We're talking about polar groups and how to identify polar groups. So what subatomic particle, if you draw an atom, we know that every atom has a nucleus, and in the nucleus are protons, okay? A lot of positive charges, okay? Oxygen would have specifically 8 protons in its nucleus. We know protons are attracted to electrons, okay? So there are protons sitting in the nucleus of every oxygen atom, 8 of them. And I'm not going to take the time to draw all 8 positive charges, but let's pretend each positive charge is a proton and it represents the 8 protons in oxygen. So each of these 2 electrons, they don't know which way to go. They're both pulled evenly by the nucleus of each oxygen atom. It's like tug of war. Neither side is going to win, okay? Now, the moment you have a bond where you have, let's say, hydrogen versus oxygen, okay? And there's 2 electrons, again, sitting in between these atoms, hydrogen, oxygen. Which way are the electrons going to be pulled? We've got 1 proton on this side, and we've got 8 protons. Okay, that's like, Cameron, you versus 8 other football players on the team in a tug of war match, okay? Who's going to win? Obviously the side with 8, right?
Right.
So these electrons are going to be pulled this way in the bond. That is polarity. So because of that, because these electrons are not shared evenly, they're shared in the bond, but they're not shared evenly. We put a partial negative on oxygen, we put a partial positive on hydrogen. We say hydrogen is electron deficient in the bond, oxygen is electron rich. They're both in a bond, and these electrons are still shared, they're just not shared evenly. The way they would be for atoms that are either identical or very similar in electronegativity. Now, I've not been using this chart because, again, this is stuff that is all covered in a previous chemistry course. But to technically be considered polar, your electronegativity difference has to be 0.5 or greater, okay, to 1.9 specifically. If it's above that, then you're so polar that you're now ionic. So when we're thinking about molecules, any molecules that have a group, a functional group, or a particular bond where the electrons are shared unevenly, that is now a polar portion of a molecule overall. So you have this idea of individual polar bonds, and now you can apply that to overall molecules, because molecules can have lots and lots and lots of bonds. Okay, we've been looking at molecules that are huge. Proteins are massive. They have tons of bonds, okay? And each individual bond, so what determines if a molecule is polar overall is going to be if it has polar bonds or not. If it has lone pairs, because that is an area of a lot of electron density. And if it is symmetrical or not. So the shape, I just say shape. These are 3 determining factors, okay? So I always walk through water as an example, okay? You look at these bonds here, we just decided, I looked at an oxygen-hydrogen bond, we just decided that oxygen-hydrogen bonds are polar because those electrons are not shared evenly, they're pulled towards oxygen, okay? So that is why, especially in these lone pairs, oxygen has 2 lone pairs, in addition to the fact that these bonds are not, in addition to the fact that these bonds are polar. So we put the negative, partial negative charges here. We put the partial positive signs on hydrogen, and we can draw in this red arrow is a permanent dipole. Okay, so that's just an example. But anytime you see, I mean, you see huge molecules in our class. You see stuff that, you know, looks like this, and maybe you got an OH group there. Whoops, that's not aromatic. Okay, maybe you've got a bromine here. Maybe you've got another OH here. I mean, it could just go on and on and on. So how do you decide which parts of this molecule are polar? It all depends on the basic things and principles outlined in chapter 4, okay? So that's why on day 1, when we went over chapter 12 and I was introducing functional groups, I said, here are some sections. Do I have that lecture here? I don't think I do. But I said, here are some sections to review. If you go back and look at one of the very first couple slides on chapter 12, I said, go back and review section 4 or chapter 4 section, I forget what section this is, but it's on that one of those very first slides from chapter 12. It really walks you through polarity, because that's a huge part of this course, is understanding when things are polar and when they're not. Okay, and I guarantee you a lot of people can struggle with it.
Okay, so the NH3 on number 1, that'd be non-polar because it doesn't have any lone pairs.
Lone pairs aren't the only thing. Polar bonds as well. You got to assess, is a nitrogen-hydrogen bond polar? And if we go look at our electronegativity chart, again, we're past the point of really calculating this out, but nitrogen and hydrogen, what's the difference there? 3 and 2.1, it's a difference of 0.9. Let's look at our table. Yep, that's polar. So because you got polar bonds, when you look at the molecule, the substrate in the lab that we're working with is right here. Okay, so what this is it's nitrogen, and then it's got hydrogen, and then it's got hydrogen, and it's got another well, that goes to the carbon, and it's got another hydrogen. And so all these bonds are polar. There's no lone pair, but we got a plus charge on it because there's 4 bonds. But all these bonds are polar as well. But again, it's multiple concepts combined. It's I don't even know if I'm answering your question at this point. What was your question?
How to find if it was polar or non-polar.
If this is polar, non-polar?
Yes.
This is ionic. Why is it ionic? Because you see the charge on it.
So it's not polar or non-polar, it's ionic.
It's so polar that it's ionic.
So in my answer on number 1, you want me to write ionic and polar, or it's so polar that it's ionic?
No, your answer on number 1, I just want you to tell me what type of amino acids would interact with that group. You're not telling me what kind of group this is. You need to acknowledge in your head, okay, what kind of group is this? Okay, if it's ionic, what kind of amino acids would be attracted to a group that has a positive charge? What kind of side chains?
Okay, thank you.
I hope that helps. This is a complex topic, okay? You're not trying to pick the groups that look like you're not just trying to say, okay, I got a positive charge here, so a positive amino acid will interact with that. No, you're saying what would be attracted to that? I hope that makes sense.
So we literally just need to put like in the where'd it go? Basically looking at these charts.
With
we
the
just need
amino
to
acid,
put- Yeah, the amino acid. We just need to put which one it's gonna interact with, not what kind it is. Like
right, so you're telling me the question here, this is the question. What type of amino acid residue would you expect in the active site of an enzyme that binds this drug? So if I were to reword this question, enzymes have an active site, we define that, and those active sites are going to contain amino acid residues, just like what we saw with the chymotrypsin today. Oh, that was on the enzyme lecture. So I'm getting to your question, Maggie.
Maggie, I just want to pop in and say, when you write down what kind it is, it makes it easier to see what it's going to interact with if you go look at the picture shown.
That's great advice. That's great advice. So for example here, just hang with me for a second, Maggie. This blue or the gray part, I don't know how it appears to you, blue or gray. This is the active site of an enzyme. The part highlighted in yellow would be like the substrate, and they come together, and then they're held together by intermolecular interactions, attractive forces, okay? So the active site of the enzyme has the amino acid serine, and there's its side chain, has the amino acid histidine, and there's its side chain, has the amino acid aspartate, there's its side chain, okay? So again, we have a totally different substrate here. We're looking at it from the viewpoint of here's our substrate, what kind of amino acids we expect in the active site. So we're given the yellow part on our lab, and we're trying to say, okay, what kind of amino acids would we imagine in the blue part? Does that make sense? In this picture, you can see both the substrate and the amino acid. You're having to imagine that over here, all right? So if I had some sort of, if this was in the pocket of some sort of enzyme, okay, let's say there's my enzyme and I got my active site. What kind of amino acid do you think might be right here that could interact with this plus charge? What kind of amino acid could be right here that might interact with this negative charge? What kind of amino acid would be right here that could interact with this OH group, okay, or this oxygen right here? What kind of amino acids would interact? Remember, opposite charges attract. So you go look at your table of amino acids. And your options are, okay, I think an amino acid with a non-polar side chain would interact with this part of my substrate. Or I think an amino acid with a polar side chain would interact with this particular part of the substrate. Or I think an amino acid with an acidic side chain or an amino acid with a basic side chain. So you got 4 things you can choose from for your answers on these questions on the lab.
I think the picture where you drew the pocket helped me a lot because I was trying to visualize like what am I attaching, and I feel like it helped.
This right here, this right here.
Yes, I think that helped me a lot.
I don't care if you scribble all over this thing if you have to.
Okay.
Okay. I have a question about the first one on the double bonded oxygen on the bottom left. So I was working it with the girls for a minute, and I was just like gonna ask, so I know it can interact with a basic side chain, a non-polar side chain, and a polar side chain. I was just like, because it's an oxygen, so it can bond to any hydrogen bond. So I was looking at the acidic, or no, it can't. Well, I was looking at the acidic, and I have on my acidic side chains, I have like CH2. So could my oxygen bond to that hydrogen in that CH2?
You're cutting in and out, Robin, but I think I heard most of it. Let me pull this
Sorry.
up. So you're looking at these acidic side chains and you're asking, could the oxygen interact with this hydrogen?
Yeah.
Anybody want to weigh in on that? That's a great question.
Couldn't it do that as long as it has the 4 bonds to the carbon?
So we're looking here, because this oxygen we know has lone pairs, right?
Uh-huh.
Are these donors or acceptors?
They're acceptors.
Are these hydrogens donors or acceptors, or neither?
Oh, neither,
Wait, why are they neither?
because it's non-polar. No.
No, it's
about the carbon
polar.
hydrogen
Are
bonds.
hydrogens, these hydrogens are bonded to carbon. These hydrogens are bonded to that carbon. Is a hydrogen bonded to a carbon ever polar? I'll prove it to you that they're not polar. What's the electronegativity difference between carbon and hydrogen? We got 2.5 for carbon minus 2.1, 0.4. According to our table, and again, I know this is stuff I've not explained in my class, but I will be furious if whoever taught you the class before you did not go over this. I guarantee you they did. That's covalent. That is non-polar. That is not polar covalent. Hydrogens bonded to carbon are never polar. They're never donors. The only way you can get a hydrogen that is positively polarized is if it is bonded to oxygen, if it is bonded to nitrogen, or if it is bonded to fluorine. So if you don't have a hydrogen bonded to one of these 3 atoms, don't even consider it in a hydrogen bond.
Oh, it can't be to sulfide either. So it can only be oxygen, nitrogen, and fluorine.
No, I mean, look at the electronegativity difference between sulfur and hydrogen. It's the same as carbon. So if carbon hydrogen bonds are nonpolar, sulfur hydrogen bonds are nonpolar.
Okay. Okay, now that helped me a lot. Okay. Now I gotta go back and fix this.
So if it's a neutral, it should be able to bond to an acidic and basic? As long as it's.
What do you mean if it's a neutral? if-
So like that double bonded O, isn't that a neutral side chain?
This one right here, Abby?
No. Yes, yeah.
So this is an oxygen carbon bond, right?
Oh.
So if y'all need to, I will put an electronegativity chart on your exam resources page. I will. I have no problem doing that if y'all need it. I mean,
That is polar neutral though, right?
it's polar neutral.
Okay, the way, okay. I was like, that's polar neutral.
It's
Okay.
carbon. It's absolutely polar. If it helps you, write in this. But here's the deal. I don't want y'all to get so like that will take time away from you on the exam if you like get so absorbed in like and it does depend on all this. These are just things that you got to be able to like. But I know it's not easy. There's a lot going on.
So Robin, what did you have on that double bonded O that she has highlighted?
What part do you mean?
Like interacting with it? Did you have?
Well, okay, so I just went back and looked. It does interact with a basic side chain. That's for sure. Because I have a nitrogen and hydrogen bond. And then I'm looking at the non-polar because I put down non-polar side chains. And it would interact with the non-polar because in
Okay, Robin, I'm so
tryptophan-
confused.
What?
I'm sorry. Okay, so you said this is going to interact with basic?
Yeah,
How?
because I have my oxygen, my double bonded oxygen, and I have a nitrogen and a hydrogen, so it can bond to that
Where's
hydrogen.
your hydrogen?
I have one on histidine.
Okay. Histidine is okay. Histidine is tripping y'all up, isn't it?
Well, and I have a nitrogen and hydrogen on arginine.
That's why I was thinking it goes to a basic.
So you're looking at this right here,
Yeah. No.
this right here. This has a plus charge. This is
Yeah,
a
I'm
big old.
looking at that one.
I wasn't looking at that one. But I mean, it has a nitrogen hydrogen, so it can bond because it's polar.
But it's considered the way we classify. It's considered basic.
Yeah. So
If it was only polar, we would just put it in the. But it's got a basic group. So the way I want you to. How do I?
so it could interact with a basic.
Well, that's why I'm questioning you. You said it could. It cannot.
It could. What? Whoa! Why can't it? Because you said nitrogen and hydrogen, and I have a hydrogen and nitrogen bond, so that makes no sense.
Yeah, so I'm thinking if it's a neutral O, it can take one of those. Oh, well, then it would be an OH plus.
Please explain to me why it can't bond with a basic.
That's what I'm gonna do.
And then I want to go. Yeah. Oh my. Okay. Oh my. brain. Oh, I'm done. I'm done because that just messed me all up.
Oh, okay. All right. Let's just slow the heck down for a minute. All right. Let me just erase everything. Robin, take a deep breath.
Oh my God. I'm so confused.
Okay. I'm glad we're talking about this then. If I can simplify this for you. Non-polar amino acids interact with non-polar groups. Okay, non-polar functional groups, non-polar parts of a molecule. Okay? Your non-polar groups are gonna be anytime you see portions of a molecule that are dominantly carbon and hydrogen only. All right? So let's go look at our substrate.
Whoa.
So like right here, we got carbon, hydrogen, carbon, hydrogen. Robin, I'm gonna get there. I wanna explain all of it,
Mhm.
okay? Look, we got this is a non-polar region. So maybe an amino acid right here would interact with a non-polar region, okay? That makes sense.
Oh.
So this is a non-polar part of our substrate. So a non-polar
Delta, quit snoring.
non-polar amino acid would interact with that. Okay, how? Hydrophobic interactions. Okay, now let's go down to the next, any questions on that? Non-polar interacts with non-polar through hydrophobic interactions. Non-polar side chains interact with non-polar parts of molecules through hydrophobic interactions. London dispersion forces are the only forces that are present in non-polar molecules. That's chapter 8. Any questions on that? Does that make sense to y'all? Give me a thumbs up. Robin, I can't see you.
Yeah.
You didn't
Yeah.
sound confident just then.
No, I'm not confident. We gotta keep going.
Well, are you confident on what I just said? Non-polar with non-polar.
Yeah, I'm confident with that. It's
Okay,
just like,
thank you. That's what I need. All right, polar. Let's look at polar. Are any of these charged? These polar groups? I'm talking about the side chains on the amino acids. So I'm going to highlight the parts I'm talking about. This right here. Is any of that charged? What
No.
about that one? What about this one?
None of
any
them
of
are
these-
charged.
Okay, none of them are charged. All right. So non-polar, which is why we also include the word neutral. Polar neutral groups are going to interact with polar neutral groups.
Is that the only thing they're gonna interact with?
Polar neutral groups on your substrate. So let's go find some polar neutral groups on our substrate. I hate this eraser, Robin.
Sorry.
Okay. Let's see if we can find some. I'm gonna try to highlight them red. Oh, you know what? This is certainly polar. We actually identified the oxygen hydrogen bond. The oxygen carbon bond. This whole group is just polar all around. This oxygen carbon bond, definitely polar. This right here is polar, but it's technically part of a carboxyl group. So I'm gonna leave that one as it is, but focus on those two in red. Those are polar groups. They're gonna interact with polar side chains.
And that's the only thing they interact with.
It's the only thing, neutral with neutral. Neutral polar with neutral polar. None of these polar groups on our substrate have charges, right? So they're gonna act with polar neutral side chains.
Well, that's where I was getting confused because when I asked you earlier, I was like, well, can they interact with like, the other groups? Like, and if they can only interact with themselves, that makes sense.
You asked me that like in lecture earlier or earlier today?
Yeah, because I was like, when we were doing the homework, I was like, well, can it interact with this side chain, blah, blah, blah. And you didn't, you didn't really want to give me the answer. So that's why you just kind of like
I
beat
was
around
trying
it.
to let
but
you
it
guys
made me
struggle. I was trying to let you guys struggle with it on your own.
Oh yeah, we
Students-
struggled.
Well,
You
here's
did.
the, and I get it. Y'all hate it. You hate it when I don't just rescue you. But I can't. I can't just rescue you. I've got to let you struggle with it on your own. I have to. Or you're never gonna learn like, and I've been there. I've been on that side of things. So I know how it feels. Okay, so don't think I'm just trying to be a meanie. But I have got to let y'all struggle with it on your own for a little bit. Okay? I do that intentionally. I'll try to lead you to the right answer, but I'm not just gonna rescue you from it. Okay, and I'm not apologetic about that either.
Okay.
The next thing I want to get to is acidic and basic. All right, So acidic and basic, let's look at the amino acids. We got a here's what I see. We got something with a negative charge. Negative charge right here. And I'm highlighting both of those, like the double bond and single bond, because there's resonance. And we haven't talked about resonance, but both of them are involved in this negative charge, even though you only see the negative on one oxygen. All right, I see a positive charge here. I'm not ignoring this whole other side chain, but the dominant characteristic of this side chain is the positive charge. The positive charge, okay? So we call this basic. Positive charges are going to interact with negatively charged groups. Why? Opposites attract. Does that make sense? Now, I know histamine, technically, the way you see it at physiological pH is not charged. Okay, so that's why when I'm looking at histamine right here, it is not charged. But just above physiological pH, so at pH 7.6 and above, histamine is charged. It will have another hydrogen plus charge. It's just that at physiological pH, roughly 50% of the population of histamine atoms in our body are neutral, and just under 50% are charged. Okay? But we still group histamine with basic side chains because at just above physiological pH, it will have a plus charge on it. So it's not uncommon to see histamine with a positive charge, but they just had to pick a way to represent it in this table, and they picked neutral. But we still consider it basic because it so easily accepts a hydrogen right here. It so easily does. Okay. Now here's the deal. Here, y'all want to know where I'm really struggling in this course? There is so much information and so much more that I wish I had time to explain to you. But y'all know we are literally busting the seams of time every single day. And I can't lecture to y'all for 4 hours a day. There's nobody would retain that. Okay. So I'm sorry. There's moments where I feel like I don't have the time to go into details like this. Unless we begin to split hairs, okay? And so that's why you're getting it now. And that's why I probably didn't go into it earlier. But that's why histamine is still grouped as a basic amino acid. And I just didn't have the time to explain that earlier, okay? Acidic side chains, they carry negative charges, so they are attracted to positively charged groups. Okay, are you still confused, Robin?
I got the positive and negative down, and I got the polar can only attract to polar neutral groups, and the non-polar can only interact with non-polar groups. I got it. They can only interact with themselves besides the acidic and basic. Those switch. Now, with the non-polar and polar, the acidic and the basic, they interact by salt bridges.
Yeah.
I know that. Now, the non-polar and polar, I'm going to my chart thing. They would, the non-polar are going to interact by hydrophobic, and the polar would interact by hydrogen?
Bonding.
Okay.
She's got it, ladies and gentlemen. Okay, whoo.
And on my table.
Y'all got me all riled up. I don't want this to be harder than it has to be for you guys, but again, I've got to let you sit and think through these things because that is literally the best way you learn it, when you learn it for yourself. I mean, I know you learn from hearing me tell you too, but it only makes sense when it clicks in your brain.
Trying to see.
So with the first one, I know we went over this in lab, but with the oxygen double bonded with the negative oxygen.
Yes, Claire.
Do you just want us to label that as a basic side chain? Or do you
I
want
want you to treat this group as one. Why? Because there's resonance here, so you'll see this drawn in, I know this is not an organic chemistry class, so that's why I haven't taken the time to draw this in. But you'll see this drawn like this, okay? Oftentimes you'll see it drawn like this with dotted lines to both. What that means is the electrons in the double bond, they can flip back and forth between the oxygens, okay?
Claire, did you? Oh, sorry. Go to
So I want you to, don't separate these two oxygens. Just think of this group as one whole and tell me, okay, what kind of side chain would interact with that group?
Okay, thank you.
You're welcome.
So this is neutral.
Will this be one of the office hours that you post?
If y'all want me to.
Yeah.
I think it might be helpful because I know Cassidy and Madison are at work.
Yeah, I would be glad to.
Okay. And then I'll let them know.
Okay,
I want to before too.
great.
But yeah, I definitely think this helped me understand it better.
Good.
Okay, so just to clarify, you just want us to put what they interact with. So put polar,
Yeah,
Great.
what type of amino acid residue do you think would be in the active site of this enzyme? In other words, Maggie, let me reword that for you. What kind of amino acid would interact with each group on your substrate?
okay, because I think me trying to label what it is what's confusing me. Like, I'm just seeing too many words on the paper and
Yeah, I get that. I agree with Robin. For me, it helps me if I label the group, Maggie. Like if I say, okay, what's highlighted in red right here is a polar group. Now I go to my table, I say, okay, what is going to interact with a polar group? Oh, a polar neutral amino acid, or an amino acid with a polar neutral side chain.
Oh, see, I'm putting interact with polar neutral side chains. Do I need to put the amino in there?
No, that's not important.
Okay.
And you could say, you could say an amino acid with a polar side chain, or a polar neutral amino acid, or you could just write polar neutral and point to that. But I'm assuming that when you tell me what's interacting with this, you're not naming the group, you're telling me what's interacting with that group.
Okay, I'm putting interact with polar neutral side chains. Is that wrong or is that right? That's wrong, ain't it?
I can't let y'all get in the habit of asking me, is it wrong or is it right? But I don't worry about the specific wording of your answer. I think your understanding, your comprehension, what you write is going to be just fine. I think you understand it now.
Okay, awesome.
Okay, so I think not about the lab, I promise. I think something else that confused me during lecture was like the inhibitions. I just couldn't really understand which was which and how to tell the difference, I guess.
Like that practice problem we did at the very end.
Mm-hmm. Yeah.
Did this little summary help you at all, Abby? This was a simplified version of everything we talked about.
Oh, I forgot about that one. I haven't even looked at that one. Completely forgot.
I'm trying to see if there's a good example from the book we could look at really quick. There's one on the homework.
Is it 1959?
Uh-huh.
Those are the ones. So I already put an answer for A.
Okay, what'd you put for A?
Well, I put irreversible inhibition.
And why did you put that?
That's what I was wondering. I don't remember.
So let's read A. It says penicillin is used to treat certain bacterial infections. Penicillin is effective because it binds to the enzyme glycopeptide transpeptidase and does not dissociate.
Oh, I put it because an active site bonds permanently, I think.
Yeah, so there's one on here that says, well, the word irreversible, right? The word irreversible indicates, that is indicative of something that you cannot undo.
Okay.
So when it says does not disassociate, that means you cannot undo it. Okay, what about B? We actually talked about B in lecture as an example.
Oh.
Is it reversible or competitive?
Both molecules can be converted to aldehydes by alcohol dehydrogenase. Ethanol is the true substrate. Okay, so both bind to the active site of alcohol dehydrogenase, okay? Otherwise, it would not say both molecules can be converted to aldehydes by alcohol dehydrogenase. That means that aldehyde dehydrogenase can bind both substrates in its active site. So which form of inhibition do more than one type of molecule can bind the substrate, but one is a true substrate and one is a, I'm trying to think of another word besides the word that would give it away.
No, I know what you're saying now.
Okay. There was only one type of inhibition in which both the substrate and then the other thing bind the active site. C, what do you think about C?
Okay, so this was, I think, let's see. I think this is where I got confused in lecture because we had talked about slowing down something. And then I think we all may have got a little confused. I can't remember.
Okay, let's read C. The antibiotic deoxycycline inhibits the bacterial enzyme collagenase, slowing bacterial growth. Deoxycycline does not fit into the active site of collagenase and binds elsewhere on the enzyme.
Oh, I know what this is. Okay.
Wait, are you sure you struggle with this?
Abby, you're gonna have to teach me.
I think I know what it is without saying the answer.
I mean, I don't care. It's homework assignment. Y'all can work together on these things if y'all want to. But yeah, you sound like you know exactly how to do these. It's not going to be any more complicated than this on the exam.
On that one, I'm thinking that it's got like a spot for it to go, but something beats it and it blocks it from going in.
So the key thing to read into here is the last sentence. It does not fit into the active site. It binds elsewhere on the enzyme. There was a type of inhibition where we talked about a specific type of molecule. We called it a regulator, binding somewhere else on the enzyme and yet affecting the rate of enzymatic the catalytic activity of the enzyme. So if you read through your summary, wait, I missed one, didn't I? On the summary. I have inhibition, I have feedback control, I have zymogens, I missed one. I don't have allosteric control. So read the definition for allosteric control. Read positive and negative, and then tell me what you think this is.
But the question for this is 19.59, right?
Yes, Claire, we're looking at
It, it only gives us 3 options to pick from, though. It's going to be like uncompetitive, competitive, or irreversible.
That's what I was gonna say, and why I'm confused.
Because I originally was thinking it was going to be one of the allosteric controls.
Oh, I see that. I didn't even read the question. What kind of inhibition, uncompetitive, competitive, or irreversible is present each of the following?
But the answer I got, it still makes sense if it says like it doesn't fit into the active site.
There is one between uncompetitive, competitive. It makes it sound exactly like a negative allosteric regulator. It's exactly what it sounds like. But if you can only pick between the 3, so there was one type of inhibition that we talked about where we said a certain molecule can bind not in the active site, but it can still slow down the enzyme.
Oh wait,
Ah.
maybe I'm confused now.
I got that.
So look here, Abby. Read through these on the slide and then tell me which one sounds most like what we're describing.
Most like C.
Most like C, yeah.
So can I say what I'm thinking?
Yeah.
I was thinking it was competitive, but then the less efficiently threw me off. When we're
Okay.
talking about
But
slowing the
also
bacteria.
focus on the part that says it does not bind into the active site. Okay, this inhibitor doesn't bind the active site. It binds somewhere else. In competitive inhibition, the inhibitor does bind the active site. In irreversible inhibition, it binds the active site permanently. Here in C on this problem, homework problem, the inhibitor does not bind the active site. So the only one of these 3 where the inhibitor does not bind the active site, there's one that matches, and it slows down the enzyme. Yeah, sorry, I didn't even read the question, or I wouldn't have even considered allosteric control because it sounds exactly like allosteric, like a negative allosteric regulator.
Can I ask about B again?
Yeah.
Yeah, I'm confused about that.
So So I was thinking since both of them could bind to it, I think I'm just getting them backwards, competitive and uncompetitive.
Okay, so let's walk through this. So here's the key things about uncompetitive inhibition. The inhibitor does not bind the active site. competitive inhibition, the inhibitor does. So right here it says it binds the active site. So it does bind the active site. It's
Okay.
competing for the active site. And the first one, it's not competing for the active site. It doesn't care if the substrate binds the active site or not. It's interested in binding somewhere else. But it affects how the enzyme functions. Okay, irreversible inhibition. Yep, it does bind to the active site. So there's only one of these where the inhibitor does not bind the active site, yet still has an effect on the catalytic activity. Like it can either slow it down or shut it off altogether.
Okay.
So on B, when it says both molecules, meaning ethanol and methanol, can be converted to aldehyde by alcohol dehydrogenase, that means the enzyme can act on ethanol or methanol. That means both of these molecules can bind the active site.
Okay. Thank you.
I need an espresso.
I have a quick question on the
Yeah.
homework. So 19.57 asks about adding an oxidizing agent. And I'm not really sure, like I understand what an oxidizing agent does, but I don't really understand how that would really, like, how that would affect an enzyme.
Okay. Let me see if I. (1957). So it's just hydrogen peroxide. And it doesn't say what kind of enzyme it is. So, yeah, we didn't really discuss that in lecture, did we? I mean, here's what you could think about, Claire. I'm not sharing my screen, am I? Let me share over to this. So we did talk about oxidizing agents such as hydrogen peroxide. We talked about them from the standpoint of the fact that they're harmful. And where I mentioned them was not necessarily enzymes, but later when we talked about antioxidants, we said we're thankful for our antioxidants because these are molecules that go around and they take care of these really reactive species such as hydrogen peroxide. Okay, so oxidizing agents tend to be very reactive. And I mean, it could be, there's all different kinds, such as hydrogen peroxide. If they're very reactive, then what could they do in the active site of an enzyme?
Would it be like, would it denature the enzyme?
Well, it wouldn't fully denature the enzyme, but if you're considering well, it also depends on the concentration. I'm tempted to throw this question out since we didn't really talk about it.
I am looking at it and I am very, very confused.
What, Robin?
I said I'm looking at it and I am very, very confused.
Yeah, y'all just don't worry about this one.
I don't remember. Yeah.
An oxidizing agent, because they're so reactive, they're good in a lot of ways, but I think I mentioned this, they're good when they're controlled. So I gave the example of fire. Fire is a really good thing when it's controlled. It's a bad thing when it's not controlled. Oxidizing agents can be the same way. Because they're so reactive, they can react with things you don't want them to react with, okay? When you think of oxidation in like, before you get to organic chemistry, you think of oxidation. Remember I talked about oil rig? Do you remember when I mentioned oil rig? Oxidation is the loss of electrons. So oxidizing agents bump into things and they transfer electrons, okay? So they react with stuff. And we don't want anything to react and mess up the state of our active site, okay? The active site is specifically designed to react with substrates, okay? So when you're thinking about the active site of some enzyme, and you might have, you know, a lysine here that has a plus charge on it. Maybe you have an arginine down here that has a plus charge on it, or maybe you have an aspartate or glutamate that has a negative charge on it. I'm just making up a random active site here. So if we have an active site that looks like this, then we don't want anything to mess with those charges. Those charges need to stay there and be there for whenever the substrate comes. And if we have an oxidizing agent, okay, H2O2 or oxygen or some other kind of oxygen come into here, especially even like a free radical, okay, an oxygen with an unpaired electron. We don't, if it gives an electron, okay, these give electrons to this, we're gonna take the negative charge or take the positive charge away. It would make it neutral, or it would make it a -2 instead of a -1. So what oxidizing agent can do is they can interact with residues in the active site and change their charges if there are charged residues, or they could create other free radicals. And we just don't want things messing with our active site. But I didn't specifically get into that, so y'all just don't worry about C. You know, what you should do, you should have your homework assignment out as I lecture tomorrow. So we might just answer these as we go through lecture. Inadvertently, you know? Not maybe necessarily every single one directly, but we're gonna talk about all this tomorrow. Hopefully. Hopefully we'll get to. Well, it's 3:26. Not 9:41. We've got a few minutes left if there's any other questions.
For
Oh.
Go ahead,
Sorry,
Rob.
Maggie. I was just gonna ask a simple question. On 1943, do you want us to put the main class or the subclass?
1943.
Yeah, I think we're just putting the main class, but I just wanna make sure.
I will accept
maybe-
that. I'll accept that.
Okay.
Oh, on the lab, whenever we're saying, like the type of bonds that they can they have more than one, or will it just be one? Like, could one like by, could one like have a hydrogen bond and do a salt bridge?
Just one. Just pick
Just
one.
one?
Yeah.
Okay.